Before I start, I should say that the veterinary school in Cambridge is a very large and diverse department. And what I'm going to talk about today, infectious diseases, makes up a large proportion of the research that we do there. But we also have a thriving hospital. We also study many other aspects of veterinary medicine. And I just wanted to do a quick plug for the fact that we have a thriving oncology unit. I just draw your attention to a pack that's out front there about our new bid to try and get a brand new radiotherapy unit going in the school. But enough of that. Infections in a changing world. I'm going to try and talk to you first about some issues to do with infectious disease from my perspective. I'm glad that Nick's talked about the really major diseases, which are the worm burden, which don't actually figure in many of the public pronouncements from people like Gates Foundation, etc. I'm then going to tell you at the end a little bit about some of the research that we do if I have time. So just to put you right in the picture, these are the things that cause infectious diseases, and these are arranged in size order. So prions are simple little proteins, cause transmissible spongiform encephalopathies, which we've been particularly bothered by in England with the BSE epidemic, etc. Viruses, of course, you'll know about. Bacteria, which is what I work on. Fungi, protozoa, and as Nick was just saying, worms. All of these things cause infections in different parts of the world. So, again, it's quite interesting. Nick and I did not collude on this talk at all, but it's interesting to note that the Surgeon General in 1967 made this pronouncement: "It is time to close the book on infectious diseases and shift all national attention and dollars to the new dimensions of health, chronic diseases." And he means cancer and cardiovascular disease and that kind of stuff. Now, 1967, I'm not criticizing him for saying that. That was probably a reasonable thing to think in those days. Antibiotics worked. New vaccines were coming through. There was a lot of optimism about killing off infectious diseases. And in fact, it was around this time that the smallpox eradication program was beginning, which was incredibly successful. However, you've got to be careful about hubris, and the author, Aldous Huxley, this is a quote from something that he wrote: "Hubris against the order of nature would be followed by its appropriate nemesis." And I just wonder whether we aren't reaping the nemesis at the moment. Certainly the public awareness of infectious disease has increased and made a lot of people worry that we are approaching some kind of a nemesis situation with infectious diseases. The diseases that this is just this whole talk is a snapshot of a very complex and difficult picture. I'm hoping that in the discussion afterwards we can discuss any other diseases, any other stuff that I don't cover here. But there are quite a few of the ancient killers are still with us. Tuberculosis, top of the list there, is still a major problem around the world, in developed and developing world. It's been highlighted in terms of the fact that it's increasingly causing problems in people with AIDS. It's also drug-resistant tuberculosis is coming through and has been coming through for quite a number of years, and it is giving us real problems. So you probably all know about tuberculosis. You probably all know about malaria too. You probably don't realize that the plague is still with us. Certainly I kind of thought that the plague disappeared in 1666, which was kind of the last big plague in London. But of course it hasn't, and it's around all over the world. Plague epidemics still crop up from time to time in different countries. Cholera is a really interesting one. And this is just repeating some of the things, or going back over some of the ground that Nick talked about. Cholera used to be prevalent all over the world, and it used to cause pandemics across the world. One of the interesting early public health pioneers was John Snow in London, who some of you no doubt have heard about. And this guy does not get enough recognition. It's an extraordinary simple piece of epidemiology, where he noted that in a cholera epidemic in Soho in London, that the cases were clustering around a particular water source, and he simply took the handle off the pump. And this is the monument to him in Broad Street in Soho. It's called Broadwick Street now in Soho, and it's just behind Carnaby Street, which is quite amusing. But he took the handle off the pump, the water supply was no longer available, and the epidemic fizzled out. And this is one of the first examples of somebody showing that water supply was responsible for carrying whatever it was that caused disease. Up to that time, people believed in a miasma theory of disease, where these diseases were just carried with bad smells and bad air. In some kind of miraculous way, it caused disease. And this is a really important discovery. And of course, it led eventually to Koch discovering that bacteria cause disease, and the germ theory of disease in general stemmed really from this kind of work a long time ago, 1840s. But back to nemesis, we have bugs which are constantly changing, and I'd just like to make the point here that human beings and animals, mammals, evolve very slowly. It takes millions of years for evolution to really work with species like us, although we have brains, so that kind of speeds up some of the processes we can deal with things with. But bugs can evolve extremely rapidly. And if you take a bug like influenza virus, for example, it makes changes in its genomic RNA every time it divides. Every time it replicates itself, it changes certain base pairs in its RNA. So these things can evolve very, very rapidly in comparison with how quickly we can evolve. Then there are new challenges. AIDS was a new challenge, of course. BSE has been a new challenge in the UK. And in the US, recently I just picked out one, which is West Nile fever, which didn't really land in the US on the East Coast until the late 90s and has now spread right across the continent. It's not a major disease problem for human beings yet. It may never be, but it's kind of an interesting example of a virus that landed and then spread a new disease. But don't forget that, this is again echoing something that Nick said, don't forget that many people globally suffer from diseases that are largely beneath the radar. So, you know, Gates Foundation and people like that around the world are pouring money into important diseases like tuberculosis, like malaria. And this is extremely important, and by no means am I denigrating that effort. But there are a lot of other diseases around there that are causing a lot of problems. And I mean, I've just picked out river blindness and schistosomiasis as worms, but Nick actually, with the geo helmets, geo worms, has obviously told you all about more important diseases than that even. But protozoa, we all know about malaria, but you know, don't forget sleeping sickness, Chagas disease, leishmaniasis, viruses. Again, I've picked out dengue fever as an example of a new and emerging problem. And bacteria, many people don't realise that Neisseria meningitidis, which occasionally causes very severe bacterial meningitis in children in the developed world, is a major problem in the meningitis belt in Africa, for example. So these diseases can tend to be sometimes beneath the radar. I also want to make the point, and this is not just because I'm at a veterinary school, I want to make the point that animal health is key, especially in many poorer nations, to the global health picture. People, again, tend to forget that it's all very well making people survive some of these diseases. But if their animals are not healthy, then you're really in trouble. So in many of the poorer nations of the world, people rely absolutely on their animals for their economic needs. So I have a friend who's doing some work on tuberculosis in Ethiopia, and he told me of an example where a village lost its oxen to a disease, and those guys had to then till the soil with sticks, essentially, and so they didn't produce much of a crop, and so they had a famine the next year. Simple as that. A village a few miles down the road, their oxen were okay, their crop was okay, they didn't have a famine. It's really at the sharp end like that with animal health in the developing world. And of course there's another issue, which is zoonoses. Zoonoses are diseases which spread from animals to people. And it's quite clear that many of the emerging infections and many of the diseases that people are getting quite worried about that crop up from time to time are very often zoonoses. There's a lot of data now that AIDS is probably a zoonosis from primates in Africa. SARS was a zoonosis. And of course, with the current avian flu scare going around, avian flu is of course a zoonosis. So we need to be very, very focused in on not just treating humans and helping humans with human diseases, but focused in on thinking about how animals and animal diseases fit into this bigger picture. Otherwise, we're not going to be doing much of a job. And this is just a slide that I borrowed from Kathy Roth at the WHO, which shows the last 10 years some of the emerging and re-emerging infectious diseases that have cropped up. And you can see there's a long list here, ranging from the extremely nasty, unpleasant, acute Ebola hemorrhagic fever virus in Africa, right through flu and other viruses. Nipah and Hendra is an important and interesting virus that's cropped up recently. Right through to the fact that we still have, as I said, have plague every now and then in India, Madagascar and Peru, etc. Hendra and Nipah are a very interesting group of viruses. And this illustrates another issue about infectious diseases in a changing world. As climate change happens, as environmental damage happens, as environments are destroyed, the ecology of these infections starts to get very interesting. Infectious diseases, don't forget, are essentially an ecological problem. Infectious diseases have to live in different environments, and I include host animals as environments in this sense as well. You and I are just another eco-niche for these bugs. They don't think that you're special or anything like that. They just live in you because you're a good source of nutrients. So Nipah and Hendra are probably viruses which live naturally in fruit bats. Now the fruit bat natural environment is being destroyed slowly but surely in these countries. And this means that fruit bats are moving ever closer towards areas of human habitation. Interestingly also, the human beings are starting to grow more pigs in facilities which are pushing into the areas of the bush or jungle, whatever you want to call it, where these fruit bats are living. Pigs are fed on all sorts of slops, and especially rotting fruit is involved. And so the fruit bats come and eat the rotting fruit near where the pigs are. The Nipah and Nipah virus especially spreads from the fruit bat into the pig. The pig spreads the virus to the people who live very closely with their pigs. And there you start to see a brand new chain of spread for a virus that never previously was seen to infect human beings. So once these two different ecologies collide, because of essentially the way the modern world is developing, those two ecologies collide and you start to see new viruses spreading from one animal to another and eventually winding up in human beings from time to time. The factors affecting disease emergence, well, I've drawn this here, wildlife, humans and domestic animals in a kind of merry dance, and I want to get this point across because, I really feel strongly that this is an important nexus that we need to think about in this area. And here's a list of a number of different factors that I've just thought about. There are many others, I'm sure, but some of the factors involved in emergence of new diseases and re-emergence of ancient killers. And all of these things are essentially things that can change and are changing in our modern world. Transportation is an interesting modern phenomenon, and this is an amusing cartoon. Be a virus, see the world. And this gentleman here is chatting up this beautiful woman in this bar, and she's from England, so he's going to go and travel to England with her and take all his viruses with him. And the point of this cartoon, which is quite amusing, is that, of course, and this is just I just put this up because I love this photograph of a 747 flying into the old Hong Kong airport through the skyscrapers. Much scarier, I think, than any infectious disease for me, anyway. But a few facts about transportation at the moment. No city on Earth is more than 24 hours away from any other. Okay? Actually, annually, the world's airlines carry some 2 billion passengers. And at any one moment, there's 500,000 people worldwide flying in a commercial aircraft. This is amazing. These figures are amazing, okay? And, there could be a new virus crops up somewhere in the world, and within 24 hours, it could be across the entire planet. Whereas previously, a couple of hundred years ago, it might never have got hold in human beings because it would just have fizzled out in the local area. Nowadays, it could be spread to a very large and wide, susceptible population, and you could get a global pandemic happening very, very quickly. And of course, we nearly had one with the SARS outbreak. SARS was an extremely serious problem. There were 8,000 patients and 770 deaths. Now, that's a very high mortality for an infectious disease. And it was the first 21st century global epidemic in the sense that it was definitely carried around by aeroplanes. And there are a few, you can't read this, but there are a few examples here of people who were called super spreaders. So there's a flight attendant here who went to Hong Kong and then went to Singapore, and then afterwards infected around 100 different people just by herself wandering around in aeroplanes around the world. And of course, we're extremely concerned in the UK at the moment about bird flu, as everybody else is. There's not a lot you can do about it. With aeroplane travel, you can, of course, the public health authorities with the SARS epidemic got stuck in very, very early on, which was very useful because they largely contained the infectious disease in the area of origin. But with bird flu, there's not much you can do about it. If a bird population, a migratory bird population, starts to carry this virus, and incidentally birds carry this virus in their guts, and it doesn't usually cause them too much of a problem most of the time, unless you get a very high pathogenicity virus. But these birds, they'll fly for thousands of miles and carry this virus around the world. So there's not a lot we can do about that. I just want to touch on influenza just briefly, because it is a pertinent topic right now. Historical writings suggest that people have known about an influenza-like disease for 2,500 years or so. Now, don't be fooled. This is not the flu that you get when you ring up work and say, I've got to touch the flu, I need to take a day off. That's not flu. Professor McConnell in the vet school, who is Scottish, explained to me that you've got real influenza when you're lying in bed and your best friend comes into your room and offers you £100 to get out of bed and you refuse the offer. Okay? And you could probably call that £10,000 and you still would refuse the offer because you really are laid out for 2 weeks. Interestingly, flu is a major killer when you get a high pathogenicity virus. In 1918, it killed probably 100 million people in that big pandemic. And that was many, many more than were killed in the First World War, of course. And it had an amazing influence on life expectancy. If you look here, the life expectancy in the USA in the 20th century went up in a straight line, really, from a life expectancy of around 45 years in the 1900. And this is a kind of distorted figure, actually. It's a bimodal distribution because, of course, that's average, and a lot of people died when they were young children in those days. So it's not quite as clear-cut as it looks. But increased World War II had absolutely no effect whatsoever. The AIDS outbreak had absolutely no effect whatsoever on this increasing life expectancy. But Spanish flu in 1918 did this to it. That's a remarkable, I always find that a remarkable figure. It's a very adaptable virus. It can change very, very quickly. One of the reasons why is because its genes are in different segments. It has 9 genes in 8 segments of RNA, 8 small segments of RNA. And so what you sometimes get is a human virus and, say, an avian virus or a pig virus or whatever, co-infect a cell in a pig, usually a pig, sometimes ducks. And when these viruses co-infect, what happens is their segmented genomes mix up in that cell, and then all different segments can be repackaged into new viral particles. Many of those viral particles that are formed are non-viable or are not a problem. But very occasionally you'll get a reassortment, unpredictable, but a reassortment which will generate a fully virulent virus particle with a different surface coat. And then you're in trouble. That's when you get a pandemic strain occurring. And this has happened to our knowledge. We started looking in 1918. Of course, this has clearly happened often in human history. But in 1918 was the first one we know about, when the Spanish flu pandemic, which was an H1N1 subtype. Both the H and the N genes changed in the 1957 outbreak, which is the Asian flu outbreak, H2N2. In 1968, Hong Kong flu came along, where only the H gene changed. That's the hemagglutinin surface protein on the flu bug. And then since then, we've had a few of these kind of viruses coming through. You can see, therefore, that these viruses are assorting and making new viruses all the time, really. And the one that we're, of course, worried about is the avian flu H5N1. And you'll see that it's been around for quite some time. And the important thing about H5N1 is that it hasn't yet acquired the ability to spread from human to human. But the scary thing about H5N1 is that just recently, the cases of H5N1 that there have been, especially in the Far East, 370 cases, 185 deaths. That's a 50% mortality. Now, 50% mortality is an extremely high mortality for any infectious disease. And if this infection did get the ability to spread from human to human and retain that mortality, then we really are in for big trouble. That's not a scare story. That's just a what if. What we're really hoping is that when it does get the ability to spread from human to human, if it ever does, then its mortality might go down. And there is some precedent for thinking that might indeed happen. And here's the map of up to February 2007 of the avian flu outbreaks, starting in the Far East and moving across. And you can see that here's our Scottish and our Suffolk outbreak in the UK that just happened just a couple of months ago. Will it make the big leap across the pond to the American continent? Who knows? What can we do about it? Well, we've got viruses, and in Cambridge at the moment there is a big Cambridge Infectious Disease Consortium based in the vet school, actually, working with a bunch of different departments and other institutes in England looking at this. And there's a guy called Derek Smith who's based in the zoology department who's doing something called antigenic cartography, which is a brand new way of trying to predict what virus vaccine strain we should use. And the WHO have just picked up on this, and they're going to use Derek's methodology to inform the choice of vaccine from year on year. We can use antivirals. Some of these new drugs like Relenza and Tamiflu. The trouble with antivirals is that the virus, as I said, mutates very rapidly, and so you get resistant virus occurring very, very rapidly. And also we have inadequate stockpiles. This is a massive stockpile in Roche's warehouse of some of these anti-flu drugs, and this is nothing like enough to deal with a pandemic. Amusingly, well, not amusing, this is an amusing thing, but seriously and importantly, biosecurity is probably a very important method to use to try and defeat the spread of bird flu. And this is actually what the Chinese, one of the provincial governments in China has put out as public education campaign. This says that washing your hands, taking the vaccine if you're offered it, cooking your chicken well and cooking your eggs well are good things to do to stop bird flu spreading. And this is where it's very interesting to compare cultures in terms of infectious diseases. These are real issues for the Chinese. Don't sleep with your chicken. Don't mix your chicken and your pigs. don't ride on your bike with your chickens, and don't slaughter your chickens at home for cooking your chicken stir-fry. So these are important messages, really. 5 minutes. Oh, jeez. I got quarter past 3, I thought I had to. Okay. Yeah, okay. Foot and mouth disease, very briefly in the UK. This is not a human disease at all, but it had massive impact on both the animals and the humans. And it was actually only had massive impact on the animals because of the human economic issues. So we want to stay foot and mouth disease free so that we can export our beef. So therefore, we have to get rid of the virus at any cost almost. And this kind of political agenda led to us slaughtering 8 million cattle and sheep in 2001 when we had a foot and mouth disease outbreak. This is how the virus started in India and moved across, again, out of India and across west up into UK in 2001. And this is what it did to our countryside. These sheep should have been dotted across the hillside, making the place look beautiful. Instead, they were in a big pile in the field here, ready to be scooped up and stuck in this lorry to be taken away and rendered. This is a very serious and quite bad thing. And it had major economic impact on the UK and on rural areas in England, which we still haven't really recovered from. This is another aspect of foot and mouth disease where farmers started to get extremely aggressive. In America, this might not be so amazing, but in England, this is pretty extreme, I can say. Although in some of the more rural areas of England, this is not that unusual. And of course, our leaders became involved. These are some of the politicians. That's the chief vet. These are some of our politicians. Chief scientist, Dave King. Lord Whitty. He went right up to our glorious leader and his amanuensis at the back there, Campbell. Our glorious leader brought in the army. And then, of course, this guy here, seen at a dinner you can't see. He's our future king. God bless him. Even he got involved in this crisis. So nothing changes because in 1865 in Punch magazine, here's a cartoon, and these are the politicians of the day. Prime Minister Earl Russell with Sir George Grey, the Home Secretary, central nervous system, where it can cause meningitis. But we don't know much more than that about it. And so what we've done recently is we've convinced the Sanger Centre to, this is the Sanger Institute in Hingston, just south of Cambridge, not far from where I live. And this guy here is a guy called Fred Sanger, who some of you will undoubtedly have heard of. He won the Nobel Prize. In fact, he won 2 Nobel Prizes. And that's who the institute is named after. And he invented DNA sequencing, basically, in Cambridge. And he invented the method that we use most often for DNA sequencing. And we're sequencing these strains of Strep suis. Their genomes are finished, actually. We've done it very, very rapidly because they're small genomes. And we're using that information to investigate the molecular basis for how these bugs interact with their natural host, the pig. And again, I want to make a pitch here that it's very important. This is from a veterinary school head, I know, but it's very important to study infectious disease organisms in their natural host if you can. I'm a great believer that it's not a good thing to do to take a streptococcus that lives in a human being and only lives in a human being and stick it into a mouse to try and understand how it works, which is how most medical research is done. That's a completely artificial host-pathogen interaction, and you will get quite a lot of artificial data out of it. So we're looking at this thing in pigs, but doing lots of infectious disease work in pigs is tricky. So especially with the the moral agenda at home. So we've invented this respiratory tract organ culture system where we take a piece of trachea from a dead animal, recently dead animal, we can chop it up into lots of bits, and then we can keep it alive for a long time. And you'll notice that these are latex beads that we put onto the surface of this piece of tissue, and you'll notice that they're being wafted off in this direction. And that's exactly what your trachea does for you. It takes particulate matter and takes it from the bottom of your lungs up your trachea, and you cough it out. And that's how you clear your lungs. And so this tissue is working in a physiological way in vitro, which is extremely useful and exciting. So the mucociliary function is maintained. We think this is a highly physiological relevant in vitro model, and we have already replaced large numbers of animal experiments by using this, which is also good for us in terms of the animal rights people at home, but it's also good in terms of economics and cost and all the rest of it. So what we can do then is we can stick our bugs onto this system and see how they interact with it, to cut a long story short. The way we screen lots and lots of mutants is by using a technology called signature tag mutagenesis, which is now about 10 years old. And we've invented a replacement for STM, but I can't talk about that now because I don't have time. But this is a much, much more efficient way of doing it. But in this particular bug, we've used STM. And what you do is you make lots of mutants in these bacteria by using a transposon, which is a piece of mobile DNA. And you can tag up each of those different mutants with a different tiny little oligonucleotide so that you can trace the fate of individual mutants in a pool of lots of mutants. So you can mix together 500 mutants or 100 mutants or 50 mutants, and you can trace the fate of each individual one of them because it's got a unique tag on it. And in this cartoon, I've shown the tags with different colours. And basically, this is the pool of organisms that you might put into your screen, into your animal or into your organ culture or whatever. And if you've got a mutant in a gene, mutation in a gene which is required for that bug to colonise that animal, it will disappear when you put it onto, that mutant will disappear from the pool when you screen it through your piece of animal tissue. And this is the cartoon version. All the bugs are there in the input pool, and there's this one gone missing in the output pool. So this flags up that this gene is important for colonising this piece of tissue. It's quite straightforward. This is what it actually looks like, not pretty colours. In this experiment, the input pool, these bugs were missing from the input pool, so we don't have to worry about them in the output pool. But these bugs that were present in the input pool have gone missing after 2 hours and 24 hours from the output pool. So this flags up that those genes are important for colonising this tissue. And these ones with the dotted lines around were there at 2 hours but have gone missing at 24 hours. So these are genes that are required not just for the initial colonisation, but for the establishment of this infection on this piece of tissue. When we did this screen, we discovered out of about 1,000 mutants that we screened, and don't forget we would have had to put that through 3,000 pigs if we'd have done it in animals. 1,000 mutants we screened on this piece of tissue. We had 17 colonisation deficient mutants, which is about the right ratio from other work that's been done. And these had transposon insertions into metabolic pathway genes, genes for microbial competitiveness, DNA replication genes, surface protein genes, and genes of unknown function. So this is now an absolute goldmine of information. But we've got to validate our technology, and so instead of screening all 1,000 mutants, we now screen just those 17 mutants. In fact, I'm just showing you a few here, through pigs in pig experiments. This is what happens when you put the wild-type organism through the pigs. All of these you get pyrexia, i.e. fever, meningitis, arthritis in these numbers of pigs, and you get colonisation of these different areas within the pig. And these are the mutants that we isolated, and you can see it's pretty stark. None of them get any clinical signs with these mutants. There's a little bit of colonisation in the upper airway, but not for very long, and none of this bacteremia or further spread into the tissue. So we are absolutely excited about this. We think that this air interface organ culture system is a way to replace the pig experiments, and is a way by which we can screen large libraries of mutants in functional genomics type strategies along these lines. What we're also excited about is that by flagging up these genes, we can now further look at these things further to see if we can make vaccines against these gene products, or if we can make live attenuated vaccines by making mutants in these things. And also, of course, the mantra is that they might also be targets for antimicrobials. I don't believe that, but that's the mantra that you always put out. That's the kind of end of that. I just want one more slide, if I may, to tell you about the Cambridge Infectious Diseases Initiative. I chair this initiative, which is a committee of the great and good in the university. Nick's on the committee, as are several others. And this is because we've recognised, of course, that infectious diseases are important. Cambridge has a diverse and excellent research base in infectious diseases that goes right across from mathematical modelling of disease through all the molecular pathogenesis stuff that I just talked about, a great deal of immunology, through epidemiology and anthropology, etc., geography, demography, all these things, all these people work in infectious disease in Cambridge. They've never really been brought together before to produce a united front to the world. We have human and veterinary infectious disease expertise, and the idea is that this infectious disease initiative in Cambridge is bringing together this extensive and diverse infectious disease community. And we want to try and get better coordination and better infrastructure, a broader appreciation of what we do, and we're also trying to engage in a high-level recruitment drive to strengthen infectious disease even further in Cambridge. And just one of the plans for a new infrastructure may include a new infectious disease institute for infectious disease research. We'll see how that goes. So that's a very brief resume of the CIDI. There's a flyer in your welcome packs about the infectious disease initiative in Cambridge. And so spread the word about that, because that's something we want to really talk up. So that's all I've got to say, apart from thank you very much for your attention.